Assalamualaikum and hi everyone. My topic of presentation is on hybridization by invasive species in aquatic ecosystem. Bull trout and brook trout are chosen for this video presentation as brook trout is the invasive species while bull trout is the native species. The two articles I'll be review are spatial patterns of hybridization between bull trout and brook trout in an Oregon stream network and changes in native bull trout and non-native brook trout distribution in the upper Powder River Basin after 20 years. To start off the presentation, hybridization is the process of producing offspring by mating two parents of different species. In order to identify a suite of markers that would distinguish bull trout and brook trout, a total of 100 learning samples collected. 30 fish is identified as the bull trout and 33 is as a brook trout and 34 is as the putative hybrid. These fish were all collected from Lake Creek. Samples were collected via electrophysics during July 2003 and identified species based on the phenotypic observation. A total of 46 microsatellite markers developed in bull trout and brook trout and identified loci that appeared to distinguish the bull trout and brook trout and to identify the hybrid. Also, the use of multiple microsatellites allowed to classify hybrid individuals as either F1 hybrid or later generation hybrid or as back-cross individual with either of two parental species. The second article of the bull trout, brook trout and bull X brook hybrid were identified based on dorsal fin bending and spotting and pattern on the back. During 2013 till 2015, they sampled the stream sample in 1990s that contained the bull trout or a mix of bull trout and brook trout. They also sampled some additional streams which are suspected to have bull trout based on anecdotal report or previous spot sampling. As for the result of the 33 fish that identified as hybrid in the group of learning samples that talked before, 24 were identified as F1 hybrid, 5 of which brook trout has mitochondrial genotype and 19 of which had bull trout mitochondrial genotype. 2 of the learning samples were identified as a back cross with a bull trout and both of these fish had bull trout mitochondrial genotype. 6 of the learning samples were identified as a back cross of a brook trout and all 6 brook trout mitochondrial genotype. 1 of the learning samples were identified as a later generation and this individual had brook trout mitochondrial DNA. Analysis of the allopathic bull trout and brook trout sample confirmed that the mitochondrial DNA marker was diagnostic between the two species. 100% of the brook trout were correctly identified in the field. 80% of the bull trout were correctly identified in the field and 87% of the hybrid were correctly identified in the field. Bull trout and hybrid were only found in middle and upper Lake Creek. The greatest number of the hybrid is 10 was observed in Lake Creek. Most of the fish sampled in Big Creek were genetically identified as trout. The majority of the brook trout in Big Creek were found in the lower end of the section. Allopathic bull trout reached the confine to 1 to 2 km at the upper end of the fish distribution. The transition downstream of the couple of kilometers through a range of mixed bull trout and brook and hybrid of the two species. Allopathic brook trout reaches the extent of much further 19 kilometers farther downstream. In bull trout stream with headwater lakes containing naturalized brook trout were found in reaches downstream of the lake and or downstream of distributaries originating from the lake. The only stream of which brook trout occur upstream of bull trout was in Lake Creek, downstream of Twin Lakes. Few exceptions is in Lake Creek, only one bull trout was found in a single ridge, and only brook trout were found at site farther upstream, where the only bull trout had previously occurred in the 1990s. A single bull trout was also found in each of the two site samples in Brook Creek, which was not sampled in the 1990s. And only one brook trout were collected in the Cracker Creek downstream of the Silver Creek. Bull trout found in several kilometers downstream of where they occurred in the 1990s. Bull trout were suspected to be in the upper Anthony Creek. However, it contained only brook trout in sample reaches that extended upstream to an 8 to 5 meter falls below Anthony Lake that would have likely precluded historic upstream migration of bull trout. In Anthony Creek and the North Fork River border current brook trout distribution extended to slightly downstream of the mouth of Anthony Creek. Data from Article 1 suggests that although the two species co-occur in all three creeks, some isolating mechanisms exist that allow the two species to maintain their genetic integrity and distinctiveness. Brook trout generally occupy the lower region of the stream, and bull trout generally occupy the upper region of the stream. The upper reaches of all three streams are located at higher elevation within a designated wilderness area, and water temperatures and habitat remain relatively pristine, presumably areas that are more favorable of bull trout. We observe F1 and Fn hybrid with both bull trout and brook trout mitochondrial genotypes, illustrating that hybridization proceeds in both directions in the Malur system. Suggests that this is the case for bull trout and brook trout hybridization event, particularly in streams such as Lake Creek, where brook trout significantly outnumber of bull trout. There are other important factors that may influence hybridization between bull trout and brook trout. The habitat in the lower section of Lake Creek has been highly modified, and temperature in section of Lake Creek are thought to be unsuitable for bull trout. The persistence of migratory bull trout has also been recognized as an important factor for the persistence of bull trout population. Although migratory bull trout spawn in Big Creek and Meadow Fork, they are absent from Lake Creek, presumably due to seasonal thermal barrier downstream of spawning habitat. The variable pattern of hybridization observed in the Malur River can be attributed both to differences in the relative abundance of brook trout and bull trout, as well as environmental conditions that may favor one species at the expense of the other. Article 2 shows that these allopathic reaches are likely key to the persistence of those populations due to the absence of hybridization and interspecific competition. Brook trout are the dominant char species in the Powder River Basin in terms of distribution and likely abundance. In Silver and Wolf Creeks, allopathic bull trout distribution extends farther downstream into slightly warmer reaches than in stream with brook trout. The lower abundance of bull trout in those reaches suggests they are less favorable to bull trout in the absence of brook trout. The narrow distribution of bull trout in the Powder River basins appear to be due to the limited availability of very cool water habitat. Fruit creeks probably does not support a self-sustaining population. The population in Lake Creek also appear vulnerable to extirpation. The reduction in distribution and low abundance of bull trout in Lake Creek is consistent with fish distribution and temperature elsewhere in the Upper Powder River Basin. Since all of the bull trout population in the Upper Powder River Basin have been limited distribution, especially where brook trout occur, those populations are more vulnerable to stochastic events, example like high severity of fires, flood and debris flows. Brook trout and hybrid distribution have not expanded since the 1990s again, with the exception of Lake Creek. Likewise, brook trout did not expand their range in most streams in the South Fork Salmon River Basin over a similar time period. Brook trout occurrence and distribution also does not appear to have changed in the Powder River basins over the course of this study. Likewise, there has been little invasion by brook trout upstream or into other accessible streams in Idaho in recent decades, and the displacement of bull trout by brook trout is not inevitable. As conclusion, through these processes, invasive species can extirpate native species, alter their spatial distribution, or change the roles of native species in food webs. Ultimately, invasive may disrupt the relationship between native biodiversity and ecosystem function. Okay, that's all for my presentation. Thank you.